Namaste to all students. Khushbu, myself Jeevan Dr. Khushbu Verma from Faculty of Agriculture and Veterinary Science. Today we are going to discuss about the transcription process, and we are going to discuss in detail each step: initiation, elongation and termination process of transcription. So basically it is part of second for transcription process, and this session is belongs to basically molecular biology program of MSc microbiology and MSc biotechnology section. So first of all, we are going to discuss about the initiation of the transcription process. So initiation of the transcription process involves the binding of the RNA polymerase holoenzyme to the promoter region of the DNA to form a pre-initiation complex or PIC. And characteristics consequences nucleotide sequence of the prokaryotic promoter region are highly conserved. If we talk about the bacterial prokaryotic promoter region, so we can understand it more by the diagrammatic representation. So here I am showing the slide where you can simply see the Pribnow box and minus 35 box. So in the Pribnow box, this is the stretch of 6 nucleotide which is Tata 80, TATA 80. So this is minus 10, or you can call it Pribnow box. And second one is the minus 35 sequence. It is a second consensus nucleotide sequence having TTGACA is centered about 35 bases to the left of the transcription site. So basically at this side minus and this side is also minus 10, and the basic difference in between minus 35 minus 10 is 17 base pair. So that's how prokaryotic promoter region showed, and the binding of RNA polymerase to the promoter region is basically followed by a conformational changes of the RNA polymerase. This is the basic first step of the transcription initiation. The first nucleotide, almost always a purine, means adenine or guanine, then associate with the initiation site on the subunit of the enzyme. In the presence of the appropriate nucleotide, RNA polymerase catalyzes the formation of a phosphodiester bond, and the nascent chain is now attached to the polymerization site on the subunit of RNA polymerase. In uh, in both prokaryotes and eukaryotes, a purine ribonucleotide is usually the first to be polymerized into the RNA molecule. After 10 to 20 nucleotides have been polymerized, RNA polymerase undergoes a second conformational change leading to the promoter clearance. Once this transition occurs, RNA polymerase physically moves away from the promoter region and transcribing down to the transcription unit and leading to the next phase of the process, which is known as elongation process. Here we can understand initiation process, and after that we are going for this initiation process of the transcription. You can simply see this is DNA molecule, and this is basically promoter region. This is RNA coding sequence. This is initiation complex which is having sigma factor. So in initiation, RNA polymerase holoenzyme first recognize the promoter region. This is promoter region, and it binds basically at the 35 minus 35 region, and basically to full the promoter, and initiation continues. RNA polymerase bind more tightly to the this yellow structure is known as RNA polymerase. It binds more tightly to the promoter region and at the minus 10 region and accompanied by a local untwisting of the DNA in that region. At this point, RNA polymerase is correctly oriented into to begin transcription at plus 1 point, which is start point. So that's how RNA polymerase start to do initiation process of the transcription. Second step is basically elongation step of the transcription process. So as the elongation complex containing the core RNA polymerase progresses along the DNA molecule, DNA unwinding must occur in the order to provide access for the appropriate base pairing to the nucleotides of the template strand. The extent of this transcription bubble, which is basically DNA unwinding, is constant throughout and is about 20 base pair per polymerase molecule. RNA polymerase basically has associated with it an unwindase activity that opens the DNA helix, and topoisomerase both precedes and follow the progressing of RNA polymerase complex to prevent the formation of superhelical complexes. Third step in the elongation process, which is base pairing rule, is followed during the incorporation of the ribonucleotide. Means ribonucleotide should be must be complementary of its template strand of the DNA. To understand about the elongation process, you can simply see the diagrammatic representation. Here you can see this is the DNA molecule. This is rewinding of the DNA, and this one is the template region, RNA polymerase, and here is the unwinding of the DNA so that this bubble can be created, and this is RNA polymerase, and then that's how RNA is going to synthesize, and this small region is known as RNA DNA hybrid region, where you can simply see that RNA is complementary binded to the DNA molecule. That's why we call it RNA DNA hybrid region, and these are the antipeas, and by the help of the RNA polymerase, these antipeas are binding and creating the new RNA strand, and these NTP arrangements will be according to the rule. Means if here is A, so in the RNA uracil will come, and if here is G, so cytosine will come. So that rule must be followed during the RNA synthesis process, and this is the elongation process. And after the completion of this region, this RNA polymerase will move forward in this direction at the 5 to 3 dash direction. And when RNA polymerase move in the direction of 5 dash to 3 dash direction, so the RNA synthesis will occur for next region. So that's how elongation process occurs. Third step is basically termination of the transcription process. So in the termination process, two steps are involved, means row factor dependent and row factor independent method. In the row factor dependent and independent method, first of all, we will discuss about the row dependent termination process. So the termination process is basically signaled by a sequence in the template strand of the DNA molecule, a signal that is recognized by the termination protein. This termination protein is known as row factor. Row factor is an ATP dependent RNA stimulated helicase enzyme that disturbs the nascent RNA DNA complex. Here you can understand the termination process, basically row factor dependent termination process. You can simply see this is sigma factor, initiation factor, and from attached to the promoter region, and then DNA RNA DNA transcription process occurs. These are the core enzymes, and this sigma factor further releases, and this is the RNA synthesized. And when termination region comes, so row factors recognize this termination signal, and then it helps to give the direction to the core enzyme to separate it so that the transcription process will be stopped here. Second type of the termination, which is row independent termination. In this process, we need requirement of the presence of intra-chain self-complementary sequences in the newly formed primary transcript so that it can acquire a stable hairpin turn that slow down the progress of the RNA polymerase and causes it to the pause temporarily. Near the stem of the hairpin, a sequence occurs that is rich in G and C content, means guanine and cytosine content. This stabilizes the secondary structure of the hairpin. Beyond the hairpin, the RNA transcript contains a string of uracil. The bonding of the uracil to the corresponding adenine in DNA is very weak, and this facilitates the dissociation of the primary transcript of RNA from DNA. That's how termination process occurs of transcription. This process you can simply understand by slide share. You can simply see in the termination region, this is RNA polymerase. These are the this is the termination signal region. So they create the RNA is having G and C content more, and at the end more uracil will be occur. So these G and C content arranged like this at the same face to face, and they create their hydrogen bonds in between, and they create the hairpin loop structure. And at the third ascent, uracil happens, and when uracil happens, so it is very weak bond in between template region of DNA and this RNA, so it easily disassociate from the DNA RNA hybrid region. So that's how termination process occurs. After the termination of synthesis of the RNA molecule, the enzymes separated from the DNA template. With the assistance of another factor, the core enzyme then recognizes a promoter at which the synthesis of new RNA molecule compresses. In the eukaryotic transcription, the general process of the transcription can be applied to both prokaryotic cells and the eukaryotic cells. The basic biochemistry of each is the same. However, the specific mechanism and the regulation of transcription differ between prokaryotes and eukaryotes. Transcription of the eukaryotic gene is far more a complicated process than prokaryotes. If we talk about the prokaryotic versus eukaryotic transcription, means if we talk about the differences in between prokaryotic and eukaryotic transcription, so first of all, basic difference is location. In prokaryotes, transcription occurs in the cytoplasm because there is no nucleus, well-defined nucleus in prokaryotic cells. But on the other hand, transcription in eukaryotic cells occurs in nucleus. If we talk about the genome size, so the genome size is much larger in the eukaryotes and greater specificity is needed for the transcription of the eukaryotic genes. Third difference is basically chromatin structure. So DNA in prokaryote is much more accessible to RNA polymerase than DNA in eukaryotes. The eukaryotic DNA is wrapped around proteins called histones to form structures called nucleosomes. Eukaryotic DNA is packaged to form chromatin, and while RNA polymerase interacts directly with prokaryotic DNA, other proteins mediates the interaction between RNA polymerase and DNA in eukaryotes. Fourth difference is RNA polymerase. There are three distinct classes of the RNA polymerase in eukaryotic cells. All are large enzymes with multiple subunits. Each class of RNA polymerase recognizes a particular types of the genes. If we talk about the RNA polymerase 1, it basically synthesizes the precursor of large ribosomal RNA 28S, 18S, 5.8S. RNA polymerase second synthesizes the precursor of the messenger RNA and small nuclear RNA. RNA polymerase third basically synthesizes small RNA including tRNA, small 5S rRNA and some snRNA. So this is also a basic difference in between prokaryotic transcription and eukaryotic, because in the prokaryotic transcription only one type of RNA polymerase is involved, but in eukaryotic transcription there are three types of RNA polymerase which are involved in the transcription process. Fifth difference is promoter region. Eukaryotic promoters are more complex in comparison to the prokaryotic promoters. Two types of sequence elements are promoter proximal and distal regulatory elements. There are two elements in the promoter proximal. One of these defines where transcription is to commence along the DNA, and other contributes to the mechanism that control how frequently this event is to occur. Most mammalian genes have a Tata box that is usually located 25 to 30 base pair upstream from the transcription start site. So these are the basic differences, and the consequences sequences for Tata box is TATA though numerous variations have been characterized. Sequences further upstream from the start site determines how frequently the transcription event occurs. Typically of these DNA elements are the GC and CAAT box, so named because of the DNA sequences are involved. Each of these boxes binds to a specific protein, and distal regulatory element enhances or decreases the rate of the transcription. They include the enhancer or silencer regions and other regulatory elements. Sixth difference is promoter identification. So, in contrast to the situation in prokaryotic and eukaryotic RNA polymerase alone are not able to discriminate between promoter sequences and other region of the DNA. The Tata box is bound by 34 kilodalton Tata binding protein, which is TBP, which in turn binds several other proteins called TBP associated factors, which is TAF. This complex of TBP and TAF is referred to as TF second D. Formation of the basal transcription complex begins when TF second D binds to the data box. It directs the assembly of several other components by protein, DNA, and protein-protein interaction. The entire complex spans DNA from position -30 to +30 relative to the initiation site. The binding of TF second D to the Tata box sequence is thought to represent the first step in the formation of transcription complex on the promoter. Another set of the protein, which is basically co-activators, help regulates the rate of the transcription initiation by interacting the transcription activators that bind to the upstream DNA elements. Seven difference is enhancer and repressors. A third class of sequence elements can either increase or decrease the rate of the transcription initiation of eukaryotic cells. Means these enhancers and repressors are basically represents in the eukaryotic genes or eukaryotic cells. So these elements are called either enhancers or repressor or silencer depending on which effect they have. They have been found in a variety of the locations, both upstream and downstream of the transcription start site and even within the transcribed portions of the some genes. In contrast to proximal and upstream promoter element, enhancer and silencer can exert their effect when located hundreds or even thousands of bases away from transcription units located on the same chromosome. Hormone response elements acts as or in conjunction with the enhancers or silencer. 7 is termination of the transcription. The signals of the termination of transcription by eukaryotic RNA polymerase second are very poorly understood. Eighth is difference is basically processing of primary transcript. Messenger RNA produced as a result of transcription is not modified in prokaryotic cells. Eukaryotic cells modify messenger RNA by RNA splicing, 5' and capping, and addition of the poly A tail. Means in the RNA synthesis or transcription process, messenger RNA can't be modified in the prokaryotic cells because they are not having any non-coding region. But on the other hand, in eukaryotic cells, they have to do RNA processing because messenger RNA is having coding and non-coding regions both. So first of all, we need to do splicing of the RNA, and then for the protection of messenger RNA from the degradation, because this messenger RNA moves or transfer from the nucleus to the cytoplasm, and in the cytoplasm RNAs will be present, and these RNAs enzymes are basically involved in the degradation of the RNAs. So when messenger RNA comes from the nucleus to the cytoplasm, so already presented RNA polymerase can attack on the messenger RNA and can degrade the messenger RNA. So for the prevention of this step, uh, nature has created the 5' and capping and addition of the poly A tail region in the messenger RNA before the transportation of messenger RNA from the nucleus to the cytoplasm. So this is the basic major difference of the processing of primary transcripts. Most eukaryotic RNAs are synthesized as a precursor in the eukaryotic transcription process that contain excess sequences which are removed prior to the generation of the mature. Means we have to remove the non-coding region, and we have to just use the functional RNA, means coding region, which can synthesize further the protein. So this is the sum up of this session, and if we talk about the major key points of this session, so you can simply understand that what is the basic difference in between RNA polymerase types in eukaryotes, and RNA polymerase second is involved in which type of RNA synthesis. And one more thing, you can understand by the transcription factors. You can understand, you have to know about the transcription factor rule. You have to know about the row factor and row independent termination process. You have to know about the DNA RNA hybrid step. And these are the important key points of this session. And this session is now sum up. And this session is powered by digital version 2.0 Jyoti Vidyapeeth Women's University Jaipur. If you have any query, then mention in the comment box. I will resolve it. Thank you.